concept 17.6, the concept of a gene is universal. In this concept, we will be discussing how the code is nearly universal. We will compare gene expression between the three domains and revisit the definition of a gene. The nucleotide for the amino acid code is nearly universal. We can take a human gene and put it either in a yeast, which is a fungus, or a bacterium, and it will make the human gene product just like a human cell would. We have put bacterial genes in plants, which totally freaks out some people. It doesn't bother me, but either way, it illustrates that the code is the same for animals like humans, fungi, plants, and bacteria. This indicates that the code came from a single ancestor cell or cell population. It makes sense, since DNA is passed from parent cell to daughter cells with a few mutations along the way. All those mutations added up to changes in gene expression and in proteins, which leads to humans, fungi, plants, and bacteria that all use the same genetic code. Well, that's simplistic, but it's good enough for an introductory course. Let's look at the differences in gene expression between the three domains: bacteria, archaea, and eukaryotes. First off, eukaryotic polymerases have a very different shape than bacterial polymerases. Archaea polymerases are more similar to eukaryotes than to bacteria, even though both archaea and bacteria are prokaryotes or don't have a nucleus. Transcription factors, which aid in the start of transcription, are different shapes too. Once again, the archaea transcription factors are more similar in shape to the eukaryotic transcription factors. Bacterial and eukaryotes terminate transcription in different ways. Once again, with what we know of archaea so far, it looks like archaea terminate their transcription in about the same way that eukaryotes do. Both bacteria and archaea have 70S ribosomes, which are smaller than the 80S ribosomes that eukaryotes have. 70S and 80S refer to the size of the ribosome. Just memorize it. Don't worry about what it means, at least not for this class. The shape of the archaeal and eukaryotic ribosomes are similar, even though they're different sizes, whereas the bacterial ribosomes are very different in shape. All of this, by the way, indicates that archaea and eukaryotes are more closely related to each other than to bacteria. On to the most obvious difference, the nucleus of eukaryotes. This allows eukaryotic mRNAs time and space to be modified. Prokaryotic mRNAs, whether in bacteria or in archaea, can be translated as soon as they peek out of the RNA polymerase, which is impossible in eukaryotic cells because of the nucleus membrane barrier. Now that we've gone over transcription and translation in detail, let's revisit the definition of the gene. Beadle and Tonham defined it as one gene equals one protein. Well, they said one enzyme, but not all proteins are enzymes. We now define it as one gene equals one gene product. The genes may code for our RNA and may code for part of the protein that will combine with other parts to form a functional protein. Think of quaternary protein structure. You may want to go reread that part in Chapter 5. The second definition is more accurate. Now I thought I'd go ahead and throw in a figure that shows a very nice summary of gene expression in eukaryotic cells. I would suggest that you get so that you can draw it from memory. But let's go over it in real brief detail, just as kind of a summary. So, in transcription, a portion of DNA that contains a gene is unwound and transcribed by RNA polymerase. We make an RNA transcript of the transcription unit. If this transcription unit happens to be for a protein, it becomes an mRNA. In eukaryotes, a cap is added. It's off to the side. You can't see it in this part, but you can see it down here. And a poly A tail are added, and introns are removed. The mature mRNA is exported out of the nucleus, and a ribosome clamps onto it during initiation of translation. tRNAs that are charged by amino acetyl tRNA synthases come in to the A site. When they move into the P site, the growing peptide chain is moved to the new tRNA. The empty tRNAs leave through the E site. In this way, the information on the mRNA is translated into a functional protein. Well, that's it for this chapter. I have a summary and reminders of what the learning objectives for these concepts are.